Welcome to the explainer. You know, your brain has this incredible superpower. It can literally change and rewire itself based on every single thing you do. Today, we're going to dive into how that works. Let's get into it.
All right, here's how we're going to break this down. First, we'll get a handle on what synaptic plasticity actually is. Then, we'll zoom way into the molecular level to see how it works. After that, we'll see how it builds actual physical memories. And then, we're going to look at what happens when this amazing system goes wrong, using Parkinson's as our main example. And that'll lead us to our final and I think most important point. Okay, so let's kick things off with the big idea. What exactly is this synaptic plasticity? And why should you even care? The answer is a huge, resounding yes. In fact, it's happening inside your head right now, as you're listening to me. This whole process, it's literally the foundation of every single thing you learn and remember. We call this synaptic plasticity, and really all it means is that your neurons can change how strongly they're connected to each other. They can even create brand new connections or get rid of old ones, all based on your experiences. You've probably heard the phrase neurons that fire together, wire together. Well, this is it. Every time you practice a new skill, learn a fact, or even try to break a bad habit, you're not just thinking differently, you are physically remodeling the structure of your brain. Okay, that's the big picture, but how does this actually happen? Like, what's going on at the microscopic level? Let's zoom in and look at the actual nuts and bolts of how these connections get stronger or weaker. So it really all boils down to one thing: frequency. If neurons are firing back and forth really fast, that's high-frequency stimulation. It strengthens their connection. We call this long-term potentiation, or LTP for short. You can think of it as your brain's save button. On the flip side, if the communication is slow and infrequent, that connection gets weaker. That's long-term depression, or LTD. It's the use it or lose it principle playing out in real time inside your head. So to really get how this strengthening works, we need to meet the two main characters in the story. There are two types of glutamate receptors called AMPA and NMDA. Just think of them as two different kinds of gates sitting on the surface of the neuron. Now what happens next is it's a brilliant two-step process. First, the neurotransmitter glutamate opens up those AMPA receptor gates, and sodium ions rush in. Now, if this happens fast enough and often enough, it changes the electrical charge of the whole neuron. And that change is the key. It literally pops a magnesium ion out of the NMDA receptor, kind of like kicking a bouncer out of a doorway. Only when that bouncer is gone can the real VIP, calcium, rush into the cell. And calcium is the master signal that kicks off all the long-term changes. And that second step, opening that NMDA gate, is absolutely critical. We know this from experiments going back decades. If you block those NMDA receptors, you basically block the ability to form certain kinds of new memories. It's that simple. No calcium influx means no new learning. The whole system grinds to a halt. All right, so we've got this tiny chemical event, this little spark. But how does that turn into something real, something lasting, like a memory? Well, it has to become physical. It has to be built into the structure of the brain. And this doesn't all happen at once. It unfolds over time. That initial rush of calcium, that happens in seconds. But then, within minutes, the cell starts moving more of those AMPA receptors up to the surface. It's like adding more antennas to get a better signal. Then, over a period of hours, something even more amazing happens. The neuron can actually grow brand new physical structures, called dendritic spines, to lock in that connection for the long haul. So you see, there are really two kinds of change happening at the same time. You've got a functional change, the synapse just gets better and more efficient at its job, and you have a morphological change. The actual physical shape of the brain's wiring is altered, and that is how a fleeting experience, a passing thought, becomes a hardwired skill or a permanent memory. Now, this whole system is just incredible when it's working properly, but all that complexity has a downside. It also means it can be a major point of failure, and that's exactly what we see in a lot of brain disorders. In fact, there's a name for this. Scientists now call a whole class of these disorders synaptopathies. A synaptopathy is basically any disease where the root problem, the very first thing to go wrong, is happening right there at the synapse. And this isn't just a theory, we can literally see this under a microscope. For example, in some forms of autism, we see way too many of those dendritic spines, and they look immature and thin. In conditions like schizophrenia and Alzheimer's, it's the opposite. There's a massive loss of them. The physical wiring of the brain is just visibly different. So to really understand how this all plays out, let's zoom in on one powerful example:
Parkinson's disease.
Okay, look at this number, -80%. That's staggering. Before the major motor symptoms of Parkinson's even show up, the person has often already lost around 80% of the dopamine-producing neurons in that part of the brain. It just shows you how incredibly resilient and good at compensating the brain is until it isn't. But here's the really interesting part. Parkinson's isn't just the disease of losing dopamine. It turns out that dopamine system is constantly talking to and balancing out the glutamate system, the one we were just talking about. So when you lose the dopamine, it throws that whole glutamate system completely out of whack. The synapses in that circuit lose their ability to change. They lose their plasticity. They can't do LTP and LTD anymore.
And you can see the results right here. In animal models, you look at a healthy brain circuit, and you get this really strong, robust potentiation. But in the early stages of the disease, that ability is just hammered. It's dramatically reduced. And by the late stages, poof, it's gone. The circuit is basically frozen. It can't adapt anymore.
Okay, so this brings us to what I think is the single most profound takeaway from all of this research on neurodegeneration. When scientists track the effects of the toxic protein involved in Parkinson's, called alpha-synuclein, they see a really clear and honestly terrifying progression. At 6 weeks, the first thing they see isn't cell death, it's that synaptic plasticity is impaired. By 12 weeks, both LTP and LTD are broken. It's not until 30 weeks or more that the neurons themselves actually start to die off. The connections fail long before the cells do. So this completely reframes how we have to think about these diseases. The crucial point is this: synaptic dysfunction isn't just a symptom of the disease. It's very often the beginning. They don't start when the cell dies. They start when the connection fails. That loss of plasticity, that's the first domino to fall. And that leaves us with a huge and I think really hopeful question for the future of medicine. If the connection fails before the cell dies, could fixing our synapses be the key? If we can figure out how to support and protect the health of our synapses, could we stop these devastating diseases before they even really start? Could we stop that first domino from ever falling? It's a powerful idea to think about. Thank you for joining this explainer.